Let's look at example 1. Predict the phenotypic outcome if a heterozygous tall, homozygous yellow pea plant is crossed with big T, big Y, big Y. Let's start by writing our legend. We know from previous examples that in pea plants, capital T is tall, lowercase t is short, capital Y is yellow because it's dominant, to lowercase y, which represents green. Let's write down the genotype of the first parent. Heterozygous tall means two different alleles for tall, so capital T, lowercase t. Homozygous yellow means two big Y's for yellow. The other parent's genotype is given. The next step is to find the gametes. So the first gamete for the first parent is going to be big T, big Y. The outer ones, big T, big Y. The inner two, little t, big Y, and the last two, little t, big Y. We're going to cross out two of them because they are repeats. We don't need to write the same gamete twice in one Punnett square. The next parent I can see is homozygous for both traits, and so it will have four of the same gametes. Big T, big Y, another big T, big Y, the inner two, big T, big Y, and the last two, big T, big Y. So let's cross out three of those, leaving only the unique gamete to put into the Punnett square. Next, draw the Punnett square, putting our two gametes from one parent along one side and the other gamete from the other parent along the other side. Next, when we put them together or fertilize the gametes to find the F1 generation, the first one is going to have capital T, big Y, big Y. The other one is heterozygous for tall, homozygous for yellow. So phenotypically, they're all tall because they contain capital T's and yellow because they contain capital Y's. Genotypically, it's a 1-to-1 ratio because this genotype is different from this one. Let's look at example 2. A dwarf red snapdragon is crossed with a homozygous tall white plant. Determine the phenotype and genotype of the F1s. Tall is dominant to dwarf. So in our legend, we'll use capital T for tall and lowercase t for dwarf. Red is incompletely dominant with white, and the heterozygous phenotype is pink. So when we're dealing with incomplete dominance, we use C for color to the power of R for red. We need two of them to code for red, CW for white, and the mix of them, the heterozygous state, is pink. Let's write the genotypes of the parents. A dwarf red snapdragon. So dwarf is recessive and red is CR. Crossed with homozygous tall white plant. Homozygous tall means two capital T's. White is CW. Next step, find the gametes. The first parent is going to have all four of the same gametes because it is homozygous for both traits. So the first gamete, little t, CR, second one, same, third, same, and the last gamete is the same. So we're going to cross out three of them, leaving only one unique gamete for our Punnett square. The second parent is also homozygous for both traits, so all the gametes are going to contain a tall allele and a white allele. So we'll cross out three of those, leaving just the one unique gamete. So in our Punnett square, we will write the one gamete from the parent over here and the other gamete from the second parent. And when we cross them, we're going to have all tall pink phenotypes and all big T, little t, CW for the genotypes. Let's look at example 3. In poultry, a crested head is produced by a dominant allele and a plain head by its recessive allele. Let's use H to represent crested head and lowercase h for plain head because it is recessive. Let's use capital B for black and lowercase b for red. Next, the genotypes of the parents. A red feathered heterozygous crested head bird. Let's start with red. It's recessive, so we have to have two lowercase b's for that. Heterozygous crest-headed means one of each allele. The second bird is heterozygous black, so big B, little b. Plain-headed bird. Plain is recessive, so both alleles have to be lowercase. Next, let's find the gametes for both parents. First gamete for the first parent is little b, big H. Second gamete, recessive b and recessive H. Third gamete, lowercase b, capital H. And finally, lowercase b, lowercase h. We have two repeats here, so we're going to cross them out and find the gametes for the second parent. We have capital B, lowercase h, capital B, lowercase h, lowercase b, lowercase h, and lowercase b, lowercase h. This time again, we have two repeats. Let's draw our Punnett square with room for the two gametes from one parent along one side and the two gametes from the other parent along the other side. Next, we will fertilize the gametes to show the F1 generation. So big B, big H, little h. Next one, we have a big B, and two little h's. Little b, big H, little h. And finally, little b, little h, little h. Let's look at the phenotypes. This first one is black and crest-headed. The second one is also black, but plain-headed. Third one is red and crested. And the last one is red and plain. The genotypes are all different, so we write 1 to 1 to 1 to 1 ratio. Let's look at example 4. A woman who has AB negative blood is married to a man who is heterozygous for blood type A, but is homozygous for the Rh factor. Determine the phenotypic and genotypic ratios of the offspring. Recall that Rh positive is dominant to Rh negative. Let's start by writing our legend. For blood type, A can be homozygous or heterozygous. Type B can also be homozygous or heterozygous. AB, we need one allele of each, and type O blood is recessive to the other types of blood, so we write it with two lowercase i's. Here we have Rh positive is dominant to Rh negative, so we'll use a capital R for our dominant trait, lowercase r for the recessive. Let's write down the genotypes for the parents. The woman is AB negative, so she needs an A and a B, and to be negative, she has two lowercase alleles for that, two recessive alleles. The man is heterozygous for blood type A, so IAI, but is homozygous for the Rh positive factor, so he has two capital R's in his genotype. What's our next step? That's right, the gametes. The first gamete for the first parent is big A, IA I should say, lowercase r. Second one is the same. Then we have IBR and another IBR. Let's cross out the repeat gametes. Let's get the gametes for the second parent. We have IAR, another IAR, little IR, and another little IR. We have two repeats here, so we'll cross those out, and then we'll draw our Punnett square with room for the two gametes from the first parent and the two gametes for the second parent. Let's now cross them. For that first one, we have IA, big R, little r. The second one, we have IB, big R, little r. Third one is IAI, big R, little r. And finally, IBI, big R, little r. Phenotypically, this one is A positive, and so is this one. So there's two A positives. Then we have AB positive and B positive. So those are our phenotypes, 2 to 1 to 1 ratio. Genotypically, they are all different because this one is homozygous for blood type A, and this one is heterozygous. And those are our 4 examples.